My apologies. Dr. Borhunter. My apologies. Thank you.
So control of peptidoglycan synthesis in Escherichia coli, better known as E. coli. A fundamental question in biology is how do you grow and divide a cell? And in bacteriology, this question is a little more complicated because the question becomes, how do you grow and divide a cell inside of a cage? Bacterial cells are surrounded by a cell wall, which protects it from osmotic stress. And this cell wall forms a contiguous cage around the cell. And it must be grown with the cell and divided with the cell, as any holes in this layer could kill the cell. Gram-negative bacteria, like E. coli, have a further complication on top of this because they have an outer membrane on the other side of that cell wall cage. And this outer membrane protects gram-negative bacteria from environmental toxins and must also grow with the cell and divide with the cell. So gram-negative bacteria must coordinate the growth and contraction of 3 different layers in order to divide. Now, going back to the cell wall, that's what I'm going to be talking to you about today. The cell wall, as I mentioned before, is of vital importance to the bacterial cell. It is so vital to bacterial cells that it is a major antibiotic target. If you want to kill a bacteria, mess up its cell wall. Our most successful classes of antibiotics, the beta-lactams, inhibit synthesis of the bacterial cell wall. So you're talking about penicillins, cephalosporins, carbapenems. Vancomycin is another example of an antibiotic which targets the cell wall, cycloserine and fosfomycin. And to give you an idea of just how devastating these antibiotics are for bacteria, I'm going to show you a video of E. coli being treated with a beta-lactam antibiotic known as ampicillin. And I want you to pay particular attention to number 3 here in this video, because it will show you what is really happening. So what you're going to see is the E. coli grow, and you're going to start seeing them pop like a balloon. But with 3, what you're going to see is the entire contents of the cell begin to bulge out of the cell wall from a hole. And that balloon of cell contents grows and grows and grows until the osmotic stress becomes too much and it pops, which gives you an idea of what that cell wall was doing, which was protecting it from the stress of the osmotic conditions outside of the cell. And the hope in research is if we learn about how the cell wall of bacteria is synthesized and how these processes are regulated, we will be able to find new targets for new antibiotics that we can use as old antibiotics become unuseful due to antibiotic resistance. So what is the bacterial cell wall? This is a micrograph of purified bacterial cell wall. This is purified E. coli cell wall. And what you'll notice is this micrograph of just cell wall looks an awful lot like those bacterial cells I showed you earlier in the video. And this is because the bacterial cell wall is what gives bacteria their shape. And it is formed of one single contiguous macromolecule which encases the entire cell. So that there is one molecule. It is not several molecules working together to form a large object. It is a single giant super macromolecule. The bacterial cell wall is composed of a chemical called peptidoglycan. Peptidoglycan is long strings of sugar, or glycans, made of repeating disaccharide units known as N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. These long glycan strands are cross-linked by short peptides, the peptido component. So peptidoglycan, long sugar chains cross-linked by short peptides. And because you have two components to the peptidoglycan, the sugar and the peptides, you need two distinct reactions to form it. The first is a polymerase reaction, which polymerizes those sugar chains. So polymerize the disaccharide building block into a long glycan chain. The second reaction you need is a cross-linking reaction, which links the peptide stems on the nascent glycan chain into the pre-existing peptidoglycan matrix. And this is how you add a strand at a time to that matrix around the cell, allowing it to grow or divide. E. coli, and indeed most bacterial cells, have two distinct types of peptidoglycan synthesis. They have the class A penicillin binding proteins. These proteins have both the polymerase activity and the cross-linking activity contained within a single protein. Then there are the SEDS protein class B penicillin binding protein complexes. These complexes have the polymerase activity in the SEDS protein and the cross-linking activity in the class B penicillin binding protein. Today, I am going to be telling you information about the class A penicillin binding proteins. And more specifically, I'm going to be telling you the story of how we learned about how the class A penicillin binding proteins are regulated. So regulation of peptidoglycan synthesis is important for survival of bacterial cells. You need to make sure that you make peptidoglycan when you need it, and you don't make it when it is inappropriate to do so. So you need something which will activate it to start it, and which will terminate it to end it. And this is what I will be discussing today, starting with the activation story, and then moving on to the termination story. To begin with, E. coli has two main class A PBPs, PBP1A and PBP1B. And these penicillin binding proteins in E. coli have a property that is very useful for genetic study of them. They form a synthetic lethal pair. What is a synthetic lethal pair? Well, what that means is under normal conditions, you have PBP1A and PBP1B, and the cells are happy and alive. If you delete PBP1A or otherwise inactivate it, the cells are still happy and alive. Same thing if you delete or inactivate PBP1B. But if you simultaneously remove the activity of both PBP1A and PBP1B, the cells will die. And this is useful in genetics because what we can do is we can start with a cell that has PBP1A and PBP1B and remove one of them, in this case PBP1A, either genetically or through chemical inactivation, and then we search for genes that become essential in its absence. So we know we will find PBP1B because it forms a synthetic lethal pair with PBP1A. If PBP1A is gone and you lose PBP1B, you die. Anything that activates PBP1B will also form a synthetic lethal pair with PBP1A. Anything required for PBP1B activity, like a cofactor, will also form a synthetic lethal pair with PBP1A, because if those are not there, PBP1B is not active and the cell will die. This genetic screen was done by Catherine Paredes-Blue in the Bernhardt lab where I was working. And a similar screen was done by Athanasios Taitas in the Vollmer lab at the same time. And what they found was that Lipo-B activates PBP1B and Lipo-A activates PBP1A. So going back, we now know that Lipo-A and Lipo-B are activating the class A PBPs. The next question then is, how? There are two activities there, the polymerase activity and the cross-linking activity. Which is it activating? One, the other, both? That's the question. And that is where I come into the picture. I wanted to know how Lipo-A is activating PBP1A. And to answer this question, I decided to take the approach of looking for mutant versions of PBP1A, which are able to be active in the absence of Lipo-A. So under normal conditions in the cell, PBP1A is only active when Lipo-A is there. If Lipo-A is not, the protein is inactive. So I looked for mutants that were active even when Lipo-A was not there. And the way I did this was to go back to that synthetic lethal property between PBP1A and PBP1B. So we know that Lipo-A is necessary for PBP1A activity. So if you delete Lipo-A, so it is not present in the cell, there is no PBP1A activity. I can then place PBP1B under control of an inducible promoter. If the inducer is present, PBP1B is produced and the cell is alive even though there is no PBP1A activity. If I remove the inducer, there is no longer any PBP1A activity and no PBP1B activity, so the cells die. Into this strain background, I can now introduce mutagenized PBP1A. And the only cells which will survive under the conditions where PBP1B is not expressed are those which are expressing a mutant version of PBP1A that can be active even though Lipo-A is not there. To give you an idea of what this looks like, if I take this strain and express wild type or normal PBP1A, you can see here that PBP1, that the cells are alive when PBP1B is present. In this assay, you take a culture of bacteria and you dilute it. And the lower the dilution it grows at, the healthier the strain is. If you then plate under conditions where PBP1B is not expressed, the cells die because the PBP1A is not active because Lipo-A is not there. If you take one of my PBP1A mutants, you can see that just like with the normal PBP1A, it is still alive when PBP1B is there. But in contrast, you can see that it is also alive when PBP1B is absent. the mutation makes it possible for the PBP1A to be active in the absence of lipo-A. Now, where were these mutations and what were they? I identified mutations all across the entire sequence of PBP1A, all of the different domains. So shown here in dark blue is the polymerase domain. In the light blue or teal, that is the cross-linking domain. The pink in the middle is the linker domain, and the orange is the lipo-A binding domain. I identified some in the cross-linker, the polymerase, and that linker domain. And we had a number of these mutations and had to select which ones we wanted to move forward with. Couldn't research all of them. And we selected G62S mutation here in the linker domain to move forward with. And the reason was that at the same time I was doing this research in E. coli, one of my colleagues, Neil Green, was doing the same sort of research in Pseudomonas aeruginosa. And he also identified the G62S mutation as a mutation that allowed Pseudomonas PBP1A to be active in the absence of Pseudomonas lipo-A. So this genetic conservation of bypassability said to us, this is probably the mutation that is getting to the core of lipo-A function. So this is the one we decided to move forward with. And the question now was, how has the activity of PBP1A changed? Because that change in activity is likely reflective of the change in activity that happens after lipo-A binding. So we did two different assays to determine the two different activities of the protein. First, we looked at the polymerization activity. In this assay, we purify the PBP1A protein, or the mutant PBP1A protein, and we give it substrate, the disaccharide subunit. And in this case, we give it the version of the disaccharide subunit from Enterococcus faecalis instead of the one from E. coli. And this is because E. coli PBP1A cannot form the protein crosslinks with Enterococcus faecalis subunit. And this allows us to specifically look at that glycan strand polymerization because there's no cross-linking happening. Once those glycan strands are polymerized, we use an enzyme to label them with biotin, run them out on an SDS page gel to separate the different lengths of strands, and then stain it with fluorescently labeled streptavidin that binds to the biotin. This allows us to visualize the glycan strands on the gel and both quantify the amount and see the distribution of length. And this research was performed, this part of the research was performed by my colleague Mario Sardis. And what he found was that this G62S mutation stimulated the polymerase activity of PBP1A almost fourfold over wild type. So it is activating the polymerase activity. Next, my colleague Marios looked at the cross-linking activity. So it took PBP1A and very similarly gave it that disaccharide substrate. But this time he used the substrate from E. coli. So those cross-linked formed because we wanted to assay the cross-linking. That peptidoglycan that was then formed was treated with an enzyme called mutanolysin, which chopped up the saccharide backbone, which left you with these neuropeptide species, just those cross-links, which could then be separated on liquid chromatography and mass spectrometry to characterize them and look for the specific chemical signature of cross-linking. And what we found was that the G62S mutation provided a modest but significant increase in cross-linking activity over wild type, although it did not approach the cross-linking activity of wild type plus lipo-A, which might explain why this mutation was only able to keep the cells alive when it was overexpressed, when there was more of the mutant PBP1A than would normally be in the cell. So from this, we concluded that lipo-A binding likely induces conformational changes in the PBP1A protein, which are transduced across that linker to activate both the cross-linking and the polymerizing activities. Separate research found that this is likely to be true in PBP1B as well. So now we know that lipo-A and lipo-B bind to the recognate class A penicillin binding protein and activate both the polymerase and cross-linking activities. The next question is, what stops? How do you terminate synthesis? A terminase is what we call the enzyme which ends the peptidoglycan synthesis activity. And a terminase is absolutely required in the cell because at the end of synthesis, these new glycan strands are still anchored into the inner membrane by the lipid tail on the disaccharide, the last added disaccharide subunit. And having the cell wall anchored into the inner membrane in that way is not good for the cell. It makes it sick. If there's too much, it can kill it. So something has to come in and cleave to separate the nascent glycan chain from that lipid tail. And a clue for what enzyme might be doing this comes in the fact that all of the glycan chains are capped by a 1,6 anhydro in acetylmuramic acid ring, or an anhydromurnac ring for short. Yeah, short. So this caps all of the glycan strands, and it looks like this. A lytic transglycosylase is the enzyme which comes in and introduces these caps. It cleaves between disaccharide subunits and catalyzes formation of that ring. So what it would be doing is it's coming in and cleaving off that last disaccharide subunit that was added to the nascent glycan chain. And that releases it from the inner membrane. You would think then, knowing this, it would be pretty straightforward to identify the enzyme in E. coli that is providing terminase activity. Unless you happen to know that E. coli has 8 lytic transglycosylases. So the question is, which one is the terminase? Well, are they all the terminase? Are some of them the terminase? So the way to approach this is to think about what are the characteristics we expect to see in a lytic transglycosylase that is acting as a peptidoglycan terminase. And those are, it needs to be able to reach the inner membrane where its substrate is located. It needs to have endolytic activity because it has to cleave within the nascent chain. And it would be expected that inactivation of what ends glycan strand synthesis will result in longer strands being synthesized. So the terminase must access the inner membrane. that nascent strand is anchored into the inner membrane. The point where you want to cleave is there. Of the 8 lytic transglycosylases, only 3 have or potentially have the ability to reach that inner membrane proximal region. MLTG, which is conveniently located in the inner membrane, SLT, which is soluble in the intermembrane space, and MLTF, which is just long enough to reach from its anchorage in the outer membrane through to the other side of the peptidoglycan cell wall. So our 8 have now become 3. So our next is endolytic activity. It needs to be able to cleave within that nascent glycan strand to release it, because the end of that strand is anchored into the inner membrane, and it cannot, as a result, chew from the end. It comes in, it cuts in the middle. MLTG has endolytic activity. This was determined by Rachel Young, who also discovered MLTG. SLT has primarily exolytic activity, but it has some weak endolytic activity, while MLTF has exclusively exolytic activity. It can only chew from the end. So our 3 have now become 2, and we're starting to lean a little more towards MLTG because it has primarily endolytic, whereas SLT has primarily exolytic. So then the last characteristic we expect to have is that inactivation leads to longer average glycan strand length. In bacterial cells, all of the strands in peptidoglycan have an average length that remains constant. Each specific bacteria has a characteristic length. So you can measure this, and then you can compare mutants to see if it changes. And when we did this, or rather Rachel did this, what she found is that MLTG have cells that are missing MLTG have longer average glycan strand length. Previous research had found that SLT mutants do not. So it's looking like MLTG has all of the properties of the glycan strand terminase. The next question is MLTG just a passive processor that comes around after the synthase is done and has left the strand and just release it? Or is it an active terminase where it is there as synthesis is occurring and helping to determine the total length of those strands? And this is where I come into the process. This is the question I was working to answer. So, active termination. What would we expect from that? Well, 2 big things. One is that the balance between the activity of the peptidoglycan synthase must be balanced with the activity of MLTG. If the activity of MLTG is too great relative to the PG synthase, shorter glycan strands are going to be made. And those shorter glycan strands will not have the same structural integrity as longer ones. And the cell will not be as healthy. And you would expect if MLTG is cleaving concurrently with synthesis, it will be in close proximity or even binding directly to that PG synthase. So I set out to test these 2 and see if they were the case. So, balance. The activities must be balanced. Otherwise, you end up with too short of glycan strands when your polymerase is impaired. I kind of came at this question sideways because I started work on a salt-sensitive strain we had in the lab that we knew had low class A PBP activity. Going back to our PBP1A and PBP1B with their activators, our strain lacked PBP1A and lacked lipo-B, but had a mutant version of PBP1B that could be active in the absence of lipo-B. These strains, this strain could grow in the absence of salt. But if you added salt and so increased the osmotic stress, the cells would not grow. They would die. So it looks like this. Our wild type strain growing under high salt conditions is fine. Our mutant strain is not. I wanted to learn more about regulation of the peptidoglycan synthesis, so I selected for suppressors, hoping to identify more activators or inhibitors that might have been inactivated. And selected for these suppressors, and this is what a suppressor would look like. It's growing, whereas before it would not. And what did I pull out? But a whole lot of inactivating mutations in MLTG. So this suggests that MLTG is antagonizing peptidoglycan synthesis. And I confirmed this by creating a clean deletion of MLTG. And then which you can see here. And then I further confirmed it by creating a catalytic mutant to make sure that it is the activity, the enzymatic activity of MLTG that is suppressing, that the loss of that activity is what's suppressing. And it was. So this suggests that MLTG is antagonizing peptidoglycan synthesis and that it must be balanced with PG synthesis. However, this isn't necessarily support the active termination idea, because MLTG we know has this terminase activity, but it could also be participating in peptidoglycan turnover. It could be recycling the PG, and that if the recycling is happening too fast relative to synthesis, that is what is impairing things. If this is the case, it would be expected that any lytic transglycosylase that participates in recycling would also be a suppressor of the salt sensitivity phenotype. And we know that many of the other lytic transglycosylases in E. coli do participate in recycling. This is previous research. So I asked the question, do any of the other lytic transglycosylases, of which there are quite a few, antagonize PG synthesis? And what I found is that not a single one did. And this suggested that it is the terminase activity of MLTG that is antagonizing the PG synthesis, that it is not a general recycling or cleavage activity that it has. So that is one point in favor of active PG termination. The second question is, does it interact with these PG synthases? To answer this question, I made use of a genetic system called bacterial 2 hybrid. In bacterial 2 hybrid, the enzyme adenylate cyclase is separated into 2 distinct domains. Each of these subunits is then fused to a protein of interest. That protein fusion, those 2 protein fusions, if they do not interact, nothing happens, and the E. coli grows as a normal white-colored bacterial colony. If the 2 proteins of interest interact, they bind to each other. This brings the 2 sections of adenylate cyclase into close proximity, which allows active adenylate cyclase activity. Adenylate cyclase then synthesizes cyclic AMP, which activates the lactoperon of E. coli, leading to production of beta-galactosidase. Beta-galactosidase then cleaves an indicator in the growth medium which produces a dye which stains the E. coli colony blue. So white is no interaction, blue is interaction. I fused MLTG and PBP1B each to a half of adenylate cyclase and looked to see whether the colony was white or blue. And as you can see here, that colony was blue and the controls were not, which indicates that MLTG and PBP1B are interacting, or are at least brought into very close proximity to each other. So the balance has to be there. The interaction is there. So MLTG has the characteristics of an active PG terminase. So that is what we think is going on. Going back to how are APBPs regulated? We have lipo-A and lipo-B, which bind and activate the polymerase and cross-linking activities. And we have MLTG, which interacts with the class A PBPs to actively terminate the production of the glycan strand. There are a number of open questions which remain in the regulation of peptoglycan synthesis, such as how are the polymerase and cross-linking domains coordinated, or are they coordinated at all? How are the SEDS BPBP synthases coordinated and regulated? And then are there organisms where the regulators or MLTG are essential, where these could be potential antibiotic targets? And if there are there small molecules that can act as antibiotics to inhibit them? Now, before we move on to questions, I want to acknowledge the Bernhardt Lab, which is where I did my graduate research and this research, as well as Rachel Young and Mario Sardis, who I worked closely with. And Mario's, some of his data is in this presentation, as well as the Rudner Lab, which is a close collaborator with the Bernhardt Lab and really helped support this research. So, any questions? Yes.
So I wonder if, if any existing known antibiotics actually do act on, like, MLTG.
No, there are not any known ones that act on it.
Maybe, and I don't really know how antibiotics, I mean, the classic story of penicillin, I don't know if most antibiotics are discovered from natural sources or now are made.
Most are natural products.
Does that kind of mean that maybe this is less likely to be a target that could, since they haven't been found naturally?
So how did I explain this? Most antibiotics have been either serendipitous discoveries, or you have something is important, and then you screen for things that inhibit it. For example, we know that DNA replication is important, and we can screen libraries to inhibit DNA replication. So part of the purpose of research like this is to say, well, for example, streptococcal pneumoniae, MLTG, and this is an actual example. In streptococcal pneumoniae, MLTG is essential. If you get rid of MLTG or inhibit it, the cells die. So now we know this, and we can go to a natural product library and say, we are going to screen for natural products that bind to and inhibit MLTG. And if we can find one, then we will go back to streptococcal pneumoniae and see if it kills it. And then if it kills streptococcal pneumoniae, then we will start clinical trials to see, is it bioavailable? Is it toxic? is it achievable at an appropriate concentration in the body? So that is actually the process by which most antibiotics are now being discovered, is you have a promising target and you then screen huge, we're talking hundreds of thousands of compounds in a library against it. So that's the end goal of this research. Yes.
So you focus on two of the bacterium. Is this likely to be able to be used on other types of bacteria as well, just not who they focus on?
That is the hope. So like I said, with MLTG in strep pneumo, it is essential. In other bacteria, different components of this overall arching system are essential, but the protein players are the same. So they all have the class A PBPs. MLTG is widely conserved. Having activators is also pretty widely conserved. So our hope is if we understand in E. coli, which is more amenable to research, what the different players are doing and how, we can then apply that knowledge to other bacterium and say, okay, I know that's important there. Let's see if it's important here, and can I target it? Yes.
So I think this is related to that. So,
Mhm.
so part of your motivation for understanding cell wall synthesis is to interfere with it.
Mhm.
You found that lipo-A is an activator for an important enzyme. Is all that right?
Yes.
But you also found mutants that don't need lipo-A.
Mhm.
So have you learned something there, like that's a path that's totally useless to pursue, or because you're figuring out what they need, but you see, it doesn't need it.
Yeah.
Have you learned something? Does that tell you something?
Yes. So one thing I can say is that was not actually easy to achieve. I had to pretty heavily mutagenize PBP1A to get those out. So maybe that's a point in favor.
So they may not; those kinds of mutations may not occur.
Yes. Well, they'll occur naturally eventually. If you have the scale of bacterial reproduction is very staggering. So they will occur eventually, but they may not occur quickly.
Okay.
But the hope here would be that you might, rather than specifically finding something that, say, prevents lipo-A from binding, you might have natural products which lock the penicillin binding protein into an active state. So by understanding what is happening when the activator binds, you're more likely to understand which parts of that protein to target with a small molecule to try to lock it off. And that might be less likely to be easily overcome. I think I have some questions here in the chat. Oh, no, those are not. Okay, well, yes.
There is a study showing the elasticity limits of the cell wall.
Yes. I could not quote you on it off the top of my head. My research was more genetically focused rather than on the biophysics of it. But that study, I definitely remember reading. It definitely exists. Yes. Yes. Yes, for example, Streptococcus pneumoniae, which I mentioned before, is a gram-positive bacteria, and in it is essential. the fundamental machinery is very highly conserved. And present in pretty much all bacteria, even some of those which don't make a cell wall. And it functions generally the same between gram-positive and gram-negative, but there are the difference, and the devil is in the details. But for example, in strep pneumo, one of my colleagues worked with that, and he was identifying a gram-positive equivalent to lipo-A and lipo-B. Now, lipo-A and lipo-B, you notice they are anchored in the outer membrane. So naturally, a gram-positive cannot have a homolog to them because it doesn't have an outer membrane. But what my colleague was finding is that even though it does not have those proteins, it did have proteins with an analogous function. So, an antibody is being introduced during the polymerization process of PBP1A affect the PBP1A activity. Well, in gram-negative bacteria, the antibodies would not have access to PBP1A and PBP1B because they are inside of the outer membrane, and the outer membrane prevents passage of large macromolecules like antibodies. In gram-positive bacteria, it is unlikely that antibodies would have access as well, because gram-positive bacteria produce a very thick, dense layer of peptidoglycan that similarly is going to occlude access of antibodies to the proteins which are underneath. So I guess to properly answer that question, no, antibodies being introduced would not affect it, at least in a cell. If you purified the proteins and put in antibodies, it is difficult to predict because it would depend on where exactly on the protein the antibodies might be binding. Yes.
Those type of glycocalyx layer on the bacteria, does it affect like those rigid or slim layer glycocalyx attached to the bacteria? Does it affect the activity of the antibodies?
So if you treat them with antibiotics, will their shape change?
Yeah.
Yes. Some antibiotics are specific to particular penicillin binding proteins. For example, in E. coli, we have, I think, cefsalodin, which inhibits the division machinery. So you get long spaghetti noodles. You have cephalexin, which inhibits elongation. So you get balls, ping pong ball E. coli. So absolutely, if you treat with antibiotics and the cell wall targeting antibiotics aren't outright killing the cells, you can get some very interestingly shaped bacteria. And in fact, in medical labs, this is something they have to actively teach your medical technologists to recognize and account for is antibiotic effects messing up the classical morphology of bacteria. And it can also lead to, I hesitate to say medical error because it's not really an error, but incorrect identification of a bacteria. And is also why, if you ever work in the hospital, the people in the microlab will constantly complain about doctors sending them samples to analyze for bacteria after they've already given the patient antibiotics, because it becomes much more difficult to recover and identify bacteria that are sick. but generally the parlance is it's sick bug effects.
More questions? Thank you so much. Good job. That was very good presentation.
Alright. Yeah. Alright, so yeah.